Okay, so we have a number of things to sort of clarify and wind up as we finish discussions of cell division. So we've gone over mitosis and meiosis. I want to cover binary fission. And this is the mechanism that is used by bacteria, so prokaryotes. All right. And the basic premise is that we're going to take an individual cell that has a single copy of its, so it's 1N, okay? It's a single copy of an individual chromosome. That's all they have. All right, it's generally circular. And what we're going to do is we're going to replicate that chromosome. All right, and it's a double-stranded molecule, but we're basically going to end up with 2 of these. All right, and then we're going to divide it. Like so. All right, so that's the basic idea of binary fission. So let's look at it here in this diagram. So we've got the circular bacterial DNA molecule here. All right, there's only one copy of it, right? So it's haploid. All right, there's a protein here that binds it to the inner membrane. All right, DNA is going to replication is going to begin, and it's going to go in both directions. All right, from that origin of DNA replication, so that start location, all right? And so it goes bidirectionally around the circle in both ways, and basically we end up with 2 copies of our chromosome, all right? Here they are, all right? We then basically create a new wall and membrane in between the two, and we have our 2 daughter cells, all right? So pretty easy, right? We don't have to do all the phases or anything like that, okay? So that is binary fission in bacteria. Now, what we've been looking at is mitosis and meiosis. So we have this, in mitosis, we have this M phase. But we haven't really talked much about interphase, all right, which is the phase in between divisions where we divide the nucleus, all right. So we have G1 phase, which is gap 1. We have S phase, which is where we synthesize the DNA. And we have G2, which is the second gap, all right. So the way to think of this is that during G1 the cell is growing, enlarging, and duplicating the duplicating the organelles, right? Sort of gearing up for growth. All right. Then it enters S phase. This is where it replicates the DNA. All right. And then it goes through a second gap that is shorter, and this is where it would kind of finish any sort of growth, verify that there aren't any issues associated with DNA synthesis, and that everything's kind of good to go forward and divide. All right. Now, if there are cells that are not actively dividing, meaning that they're not kind of proceeding through interphase to the idea of dividing, we call those G0. All right, and they are not actively dividing. So if something is still progressing through these interphase subphases, right, G1, S, and G2, it's not G0. All right. All right. So now this is a karyotype. And basically it's a description of the chromosomes in an organism. In this case it's humans. All right. So there are 23 sets. And so what we're looking at here is the fact that this organism is diploid, right? And so we've got 2 copies of each chromosome here. All right. And the basic nature of the information on each of these chromosomes is relatively the same, right? So if let's say that at this location right here, okay, so I'm at chromosome 5 and I'm making some red. So let's say that those 2 locations right here and here, they might both carry a gene for, let's just use eye color. All right. And they might differ in that one of them might be the gene to produce blue eye color pigment and the other might be to produce brown eye color pigment, right? So they're not identical, but they're similar, all right? And that's what the idea of homologous chromosomes are, except for the sex chromosomes. Now this individual is XX, all right? If it was XY, this chromosome here would be kind of shriveled up. There wouldn't be nearly as much information on it, okay? All right, so that's karyotype. Now, What we really want to talk about today is cell cycle regulation. Right? How is it that cells know they should divide, right? What triggers the divide? When shouldn't they divide? All right? So what we want to think of is that for a cell to undergo division, whether it's meiosis or mitosis, the environmental conditions around that cell need to be appropriate for growth, right, for division. And so the environment is often going to dictate what is going to happen, all right? The division is regulated by a series of proteins called cyclins and cyclin-dependent kinases. All right, so kinases are proteins that phosphorylate things, either themselves or other proteins. All right, and so what's going to happen is, if we look at this, here's my cyclin. All right, and so it's increased in concentration. It binds to a protein called a cyclin-dependent kinase, which means that this protein can't phosphorylate anything unless it's bound to the cyclin. All right? So the cyclin is produced, it binds, so now we have an active CDK. So now we have an active cyclin-dependent kinase. All right? It interacts with the target protein, it phosphorylates that target protein. Now we have an activated protein that will promote cell division. All right? Once we've sort of accumulated these proteins that are causing cell division, the cyclin degrades, is broken down, and that gives us our inactive cyclin-dependent kinase. As we move through the cycle further, cyclin will be produced again for the next round, and we'll do it again and again. All right? Okay, so let's look at what are called checkpoints. And the idea behind checkpoints are that there are points in time when you want to stop and evaluate whether or not division should occur. So let's say that a cell has the inappropriate number of chromosomes, or there's too many copies, or certain regions of DNA have been replicated too many times, so you have too much. Those things are going to have adverse effects on the daughter cells, and therefore you want to stop and repair or stop and destroy this cell that may cause issues. So if we look at some examples where these cyclins interact, all right, so here we have the G1/S cyclin CDK, all right, and it basically prepares the cell for DNA replication. So it's going to control the production of things like histone proteins that we're going to have to wrap the DNA around, all right? And so the cyclin then, right, CDK complex, it is controlling that production of what is needed before you move into S phase from G1, all right? Now, the S-cyclin CDK, so this one's different, all right, S-cyclin CDK, this helps initiate DNA synthesis. So there are basically start locations on a chromosome. So we've got a chromosome here, all right, and let's say that there's a start here, and it goes, you know, in both directions from this, right, and here, all right. Now, you don't want to start again at this same location, right? You don't want to duplicate this more than once. And so what happens is that the S-cyclin CDK, it prevents DNA polymerase, the enzyme that's going to make the DNA, from starting here a second time. So it's going to block it. All right? So that's our S-cyclin CDK. All right? Now, M-cyclin CDK helps prepare the cell for mitosis. In this case, it's going to help with the actual division. Some of these activities are phosphorylating proteins involved in the synthesis of the mitotic spindle apparatus, phosphorylating proteins that are involved in the breakdown of the nuclear envelope, all right, because we have to destroy that during mitosis. So that would be some of the things that the M-cyclin CDKs regulate and control. All right, so let's look at some specific checkpoints. So here at the bottom, this is the G1 checkpoint, all right, before entering S. And here the question would be is the DNA damaged? And if there isn't any damaged DNA, All right. Then it'll proceed to S. If there is damaged DNA, then it'll either be repaired or we'll boot this thing out to G0, right? So those are our two options, all right? So let's assume then that everything is going smoothly. So the DNA is not damaged, so we enter S and we go through S, okay? So we exit S and we're in G2. Well, now we have a G2 checkpoint that basically is going to ask the question, is all the DNA replicated, right? Do we have 2 copies of everything? Is there a sister chromatid for every chromosome? So before it will exit G2 and enter M, that has to be verified. So that's the G2 checkpoint. Now, another is the spindle assembly checkpoint, right, before anaphase. And at this point, before you start to drag those sister chromatids apart from each other, right, you need to make sure that the spindle apparatus is actually attached, right? So all chromosomes, and you may even want to think of this as each chromatid is attached to the spindle so that you can pull those sister chromatids apart, forming the chromosomes. All right, and so that's another checkpoint. All right, and so all of this, I think, starts to make sense that you want to regulate things and make sure that everything's okay before you take off. Now, this is an example of how we can turn off or shut down the cycle, okay, or turn it on, all right? So what we have is protein P53. P53 generally is produced in the nucleus, exported from the cell, and degraded, all right? Now, if there is DNA damage, okay, what will happen is that the presence of the DNA damage is detected. That will cause P53 to become phosphorylated, all right, which does not allow it to be exported from the nucleus. And the phosphorylated P53 then turns on genes that inhibit the cell cycle. So it's going to, it's basically going to attach the DNA here, and it is going to basically cause genes to be turned on that prevent the cell cycle. And so basically we will not exit G1, which is where we're we're answering that question of, is there DNA damage? All right, so pretty slick system. And so why are we so interested in the cell cycle, like what controls it? All right, so if we think about cell cycle control, all right, we're really thinking about things like cancer and proper development, right, so that we end up with proper amounts of cells in the right places and that they function properly. All right, so those are some of the key kind of applications of understanding the cell cycle. And so in terms of cancer, right, this is the uncontrolled growth of cells. All right, and in terms of developmental biology, right, it is this idea that you only want to produce kidney cells to your kidney when you need additional cells, and so you have to basically regulate when you add cells and when you slough cells off. So let's think about mucous membranes, all right, the skin. You constantly have to generate new skin cells, but you only want to generate them at the rate that you lose them, right, so that you don't end up with big, thick layers of skin, okay? And so that's why we learn about the cell cycle, when we're going to do mitosis, when we're going to do meiosis. And I'm going to leave more of the specifics on cancer for later in your biology career.